Good morning and good afternoon. Thank you for being here. My colleagues, David McNally, our Head of Process Development for Viral Vectors, and Rashmi Prasad, our Analytical Scientist in Quality Control, and I are very happy to share with you our experience leveraging our vaccine manufacturing and quality control platforms for new modalities. I will start the presentation with a brief introduction to MassBiologics for the purpose of context. Then I will move into the core topic and I will present how we are leveraging our quality systems and our facilities. Then David and Rashmi will present our approach to product process and analytical development. I will close the presentation with a summary and conclusions. MassBiologics has over 125 years of history. In 1894, the state laboratories were formed by the Massachusetts Department of Public Health. Of great pride to the organization is holding FDA license number 64, issued in 1917 for the production of smallpox vaccine and diphtheria antitoxin. During the first 50 years, our focus was on equine antitoxins. In 1946, we became the state laboratory for plasma fractionation, with a shift in focus to production of therapeutics from human serum. In 1997, the laboratories were transferred to the University of Massachusetts Medical School, and we became MassBiologics. During the 90s and 2000s, we continued to produce vaccines, but started to produce human monoclonal antibodies. In 2014, our cGMP vector manufacturing center was launched for the production of viral and plasmid vectors for gene therapies, and we initiated the use of single-use technology. In 2019, we broke ground for the construction of our oligonucleotide suite with the purpose of translating the UMMS therapeutic targets and also for contract manufacturing. A couple of pictures from our facilities. On the top and the lower left is our Mattapan site, which is in proper Boston. The top picture is our research and administration building, and in the same site, we have the manufacturing and quality control for the large scale. On the bottom left is our South Coast facility, is the vector manufacturing in Fall River, Massachusetts. Today, we operate in three core areas. First, advancing our internal pipeline of monoclonal antibodies, either as a pre-exposure prophylactic against Lyme and ETEC, or as a therapeutic product for diphtheria and COVID-19 infection. As a CDMO, we support the industry with services developing and producing antibodies and other recombinant proteins, viral vectors, reagents, and a full suite of fill-finish operations. Our oligo suite is completing commissioning and validation, and we expect to offer services mid to late 2022. Our industrial operations are anchored by commercial production of our TD vaccine and through licensing activities for Simplava, our licensed C. diff monoclonal antibody, and Ravishield, a monoclonal license in India and Thailand for rabies treatment. A couple of examples of our activities. Earlier this year, we started the clinical trial for our Lyme PrEP, and we are pretty excited with the results so far. Below, you can see a picture of one of our humanitarian efforts with our team ready to ship our TD vaccine to Haiti. On the CDMO side, on September of 2020, we announced the commercial agreement with PTC Therapeutics for production of their gene therapy product. And just last month, we received EMA approval for commercial manufacturing, testing, and release of gene therapy products at our South Coast facility. Okay, with MassBiologics in context, we will go over our approach for leveraging our systems between vaccines and new biological modalities. A sound quality system is the first enabler for moving a new modality into the manufacturing and quality control environments. Our quality systems were designed to support our FDA licensed vaccine, and except for a few alignments that were well managed within the system itself, Obtaining the EMA certificates for our gene therapy facility was a pretty straightforward activity. Likewise, having a system that is designed to support the different requirements between a commercial and a clinical product has been critical in allowing us to work across different types of products in the biological space. Some examples on how our quality system is supporting our operations. At the Matapan site, we are a licensed facility for the formulation, fill, and labeling of an FDA-licensed recombinant influenza vaccine. We have supported multiple INDs for phase 1-2 of monoclonal therapies, and the next steps for us are large-scale production of viral vectors, so approval of the facility for this, as well as approval of our oligonucleotide facility. On the South Coast, we have submitted a number of INDs for multiple gene therapy products, and of course, our commercial manufacturing of a gene therapy product for the treatment of AADC deficiency. Next is in collaboration with our partner to support the gene therapy product approval. On the facility and equipment side, the good news is that the facility fit approach that is typically used during traditional tech transfer works very well when moving between the biological modalities. Two key considerations. First, assemble an experienced process engineering and manufacturing team. It will be very, very helpful. And second, leverage and trust the feedback from your vendors. A facility fit starts with a gap analysis between your capabilities and the process. Invest on this step because it will help to avoid surprises later on. Out of the gap analysis comes a punch list of activities. Here it's important to start as early as possible and be ruthless between what is a must and what is a want. During the execution, be ready for changes, and most importantly, be open. Sometimes generating some supporting data goes a long way versus implementing a complicated and lengthy facility change. The output of a methodical and documented facility fit will be a valuable asset for many years to come, starting with the supporting a regulatory agency pre-approved protocol for moving between these modalities. This pre-approved protocol, it really de-risks the impact of activities on your commercial facility. A few examples of the documents that you will generate during your facility fit, anywhere from process reviews, outlays of your facility and the flows, list of your calibrated and validated ranges, what are the gaps and where you need to go, your punch list, and the overlay of your project timelines to your facility fit timelines. Fill-finish operations present an interesting challenge between modalities. For vaccines and antibodies, our commercial line is designed to process up to 250,000 doses per lot. Here, high capacity, high speed are the critical aspects of a commercial readiness. On the other side, in products for gene therapy, typically require less than 100 doses per lot, and many still require rapid transition to ultra-low temperatures. Here, small lines, single-use components, and minimum light losses are the critical aspects. Our small line in South Coast is validated to meet many of these needs. As the new modalities mature and product indications expand, these two capabilities will eventually merge. Next, my colleague David McNally will present our approach to process development.
Thank you, Mireli. So let's start by comparing viral vectors to more traditional protein biotherapeutics such as recombinant vaccines. To start with, viral vectors are much larger. Adeno-associated virus is around 30 nanometers in diameter and lentivirus is around 100 nanometers. Both of these are complex assemblies of multiple proteins and nucleic acid, and in the case of lentivirus, a lipid envelope. Understanding the physical characteristics is critical in the development and manufacture of these molecules, and we can leverage our experience analyzing and manufacturing vaccines to inform analysis and manufacture of viral vectors. So we don't just need to compare the products themselves, but also how they're developed. Traditional biopharmaceutical, we're all familiar with, has a very familiar development cycle. We have preclinical, phase 1, phase 2, etc., all the way to post-marketing. And the molecule proceeds through each of these phases sequentially, and the analytical methods and processes develop concurrently. For viral vectors for gene therapy that frequently target very rare indications with unmet medical need, the development cycle is accelerated to meet this need. For example, we can even skip both phase 2 and phase 3 trials. So the development of analytical methods and processes must be accelerated as well. So this requires leveraging existing expertise to enable the rapid development and implementation of commercial processes. So let's discuss some examples of what can be leveraged in the viral vector space from the vaccine industry. Let's start with an example from expression systems. Insect cells have been utilized to produce a variety of vaccines, for example flublok. The same expression systems have been utilized to manufacture adeno-associated viruses for gene therapy. Similarly, unit operations and equipment can be utilized as required, as most viral vector processes use unit operations familiar to vaccine and recombinant protein manufacture. There is a lot to leverage in the process realm. However, the technological maturity and target indications for gene therapy vectors pose challenges while highlighting the opportunities for industrialization. For example, our existing 2,000-liter manufacturing suite, we can generate in a single batch recombinant protein for a huge number of doses. However, for a viral vector indication, with current low productivities and low yields, we only generate enough material from a single batch to treat 16 to 20 patients. This is tenable as long as we can accept high cost of goods. However, as gene therapy expands into larger patient populations, the need to industrialize and leverage existing biopharmaceutical experience is critical. As I alluded to in an earlier slide, we can leverage unit operations we are familiar with for viral vectors. For example, in AAV processing, the majority of process-related impurities are removed during affinity chromatography, a step familiar to many different biopharmaceuticals. Likewise, product-related impurities can be controlled using ion exchange chromatography. Utilizing experience gained in processes for other biopharma products allows for rapid development. So let's look at a more detailed example. Let's look at one of the more complicated processes in AAV manufacturing, and an example of where we need to leverage existing expertise and experience in a unit operation. Adeno-associated viral vectors have a variety of product-related impurities, and they're best exemplified by the variety of viral capsids that can be produced. These include full, empty, and partially filled capsids. These need to be characterized and controlled, and this is frequently performed using familiar unit operation, ion exchange, where we can utilize shallow gradients or shallow steps to separate the various species. So our process experience with ion exchange chromatography allows us to rapidly develop this step. Now let's move on to methods. Potency methods in particular are critical to any biopharmaceutical product. MassBiologics has experience with a variety of potency assays. Our legacy TD vaccine is dependent on a validated in vivo potency method, which is complicated, slow, expensive, and of low precision. Likewise, we have experience with more modern plate-based biochemical assays that allow for rapid and precise measurements. We had to leverage our experience with both complicated and more straightforward biochemical assays to generate potency methods for viral vectors, which due to their complex in vivo function require complicated multi-step methods.
Thank you, David. I will now shift your attention towards the analytical and the quality control aspects of the program. Here you can see a snapshot of the important analytical characteristics of the AAV products And based on these characteristics, we designed the set of release assays, which are also used for assessing stability and lot-to-lot consistency later on. So, being a viral product, which is a gene sequence encoded in a protein capsid coat, we need to assess whether the capsid characteristics are right, assess the vector titer based by qPCR, microbiological tests assessing purity and safety, the full and empty capsid ratio, the stability by aggregation or DNA degradation, and most importantly, the potency, which measures the transgene function by a relative potency assay, and the infectivity, the infection capability of the AAV transgene by the TCID50 assay. All analytical methods are very important and are the key for the success in drug discovery, development, and commercialization of any product. Specifically for gene therapy or the new modality of the products, where the product development life cycle is often compressed due to a timeline crunch, we need to accommodate the same in the analytical method development as well. Therefore, if you correlate how a product moves from the preclinical to the late clinical stage, the analytical methods also need to move in their development, qualification, and validation accordingly. For example, at the early clinical stage, we should start thinking about what the methods are going to be and think about initial development. By the mid-pro stage of the program, there should be prototype methods that are beginning to be used in process development. It is okay to have report results only at this criteria, fix the basic physical parameters and not have specifications. By the mid stage, when the analytical assays move to the quality control space, that's when they start getting qualified for early phase GMP use. As the assays get refined, we are in a position to narrow down the acceptance criteria and set the assay as robust as possible. It's time for it to be validated when the product is in the late clinical stage. Potency assay sits at the core of product development for vaccines, monoclonal antibodies, and gene therapy. It is used for product characterization in the early stages by understanding the mechanism of action, and in late stages by actually evaluating if the product that has been made is biologically effective or not. This is used for a lot release, for assessing the stability of the drug throughout the program. It is also used for comparing different lots of the product and all the processes. In many programs, potency assay is used for dosing purposes as well. Understanding the importance, if we look at some of the desired characteristics of the assay, it needs to be sensitive, it definitely needs to be accurate and quantitative. So the challenge in potency assay development is that there are no platform methods available, since this is the assay which is dependent on the mechanism of action of the biologic, be it AAV2, vaccine, or monoclonal antibodies. It has to be developed on an independent case-by-case basis. These are often long and tedious procedures spanning from 1 to 2 weeks, and because of the cell-based components come in with a large variation. What are the approaches that can be used to develop a potency assay? The first one is an animal-based assay where we measure it directly in the living system. This is an in vivo animal study which has been used historically for our legacy products such as TD vaccine. It measures the biological activity directly but comes in with high variability and is a difficult assay to validate. Therefore, in the current situation, it is not considered a very good approach. The next is a cell culture based assay where we use the power of immortalized cell culture systems to mimic the in vivo biological function, and therefore we have the advantage of using a very well characterized cell bank system. It is often a multi-stage assay and can be a long tedious process because of the same. It is however best suited for the AAV products. The third approach are biochemical assays such as ELISA assays, often used for monoclonal antibody-based programs. And sometimes we also use a matrix-based approach where we combine two or more of these pathways. The case study, I will give you an example of a gene therapy AAV2 product. Let's call it Project X, which generates the gene product X. This was again developed and manufactured in MassBiologics and was on a fast timeline. When the assay was ready to be transferred from PD to QC, the SOP along with development reports were transferred. The limited data that was available was also transferred. There was no time to generate more preliminary data in QC. The assay had to move to validation phase, which was completed with for validation report, and based on the report, the SOP and the acceptance criteria had to be revised. These two often happen separately, one after the other, but in this case, we had to merge the two. The assay that we developed was a dopamine induction assay and spans over 2 weeks to be executed. We use HEC293 cells seeded overnight, and the next morning we add our product of interest at 7 increasing MOIs from 200 to 0.56 particles per cell. It is incubated for 24 hours, after which a chemical is added to this system, which is a substrate for this product X. Keep in mind, X is an enzyme. Therefore, when we add the substrate externally, it would be converted to its own final reaction product. This happens in the next 2 days when the substrate and the product are incubated together, after which the cell supernatant is harvested and frozen. This completes the cell culture portion of the assay. The supernatant is next estimated by a commercially available ELISA kit, and that generates the potency parameter EC50. The dopamine induction assay was validated in the QC lab following ICH guidelines, measuring precision, which measures the overall variability of the assay. Repeatability, which is an intra-assay variability, yielded about 25% CV between the results. However, the intermediate precision, or between different analysts over different days, yielded about 44% variability. Please note that such high numbers are not uncommon in the field of cell-based potency assays. Linearity was assessed over different concentration ranges of the product. We tested them from 1 to 50-fold diluted product, and we got a very good linear response, the R-square being 0.99. To establish specificity, we used our gene product of interest, which is AAV gene X, and the assay met all the acceptance criteria. However, when we used a generic reference standard, which was AAV2 GFP, we did not get a reportable result. Testing robustness over 2 different ELISA kit lots, the variability obtained was about 40%. This took 3 months, 18 assays, and 3 analysts, but the assay was successfully validated.
To summarize our presentation, the rapid growth and diversification of biotechnology products present unique opportunities. We hope that this presentation shows how the similarities between the modalities can be capitalized and play an enabling role on this growth. Of course, the suitability of manufacturing and quality control platforms to support different biological products is going to vary. An in-depth understanding of the nature of the products is critical to maximize the value of the assets. Nevertheless, we truly believe the new modalities can greatly benefit from the systems built to support legacy biologics, and to the degree that we can leverage these systems while we're investing on developing new technologies, the faster and the more impactful the results will be. MassBiologics' success was built over generations, pursuing one of the most noble of human endeavors, to heal. As an organization, we demonstrate the amazing power of combining the strength of government, academic institutions, and industry to deliver on the promise of science to improve the human condition. We thank you for your attention and look forward to meeting you in person.